get started. Today, I'm pleased to introduce Carter Hoffman, who will be presenting his thesis proposal defense. Carter is a master's student in the Bioinformatics and Computational Biomedicine major in DMICE. He received his bachelor's in zoology from the University of Washington, where he became fascinated with genetics and developmental biology. Since then, he has worked as a molecular biologist for both academic and biotech industry labs in the fields of infectious disease, cancer, and ecology. He has worked with a variety of different organisms, including viruses, bacteria, insects, small rodents, and humans. Through his research experience, he began spending more and more time with computational applications to solve the research questions he was working on. And this naturally led to an interest in bioinformatics and computational biomedicine. His current area of research is focused on strategies to identify the human bladder microbiota.
Okay. Okay, I'm not going to kid myself. I could go right to my specific aims and methods, and you'd be fine with that. But there is a lot of background that was involved with getting to those specific aims, and it's best if I explain that all to you. So I'll be doing a brief introduction of the microbiome, and then going to some of the sequencing strategies for the study of the microbiome, and then explain some characteristics that make for good marker genes. I don't think it would be a surprise to anybody to realize that there are microbes that live in and on our bodies all over, from the backs of our ears to between our toes. The Human Microbiome Project was one of the first large-scale studies to survey the bacteria from all these body sites, some of the body sites, as a start to find out what the role of this microbiota could do to the health of humans. And from this, they discovered that there's a wide variety of bacteria from all over. Different body sites have different population makeup, and that was more of a surprise than anything else. Other body sites that could have been done were left for later work, but one exception was the human bladder, which has been always considered to be a sterile environment. And I remember when I was volunteering in the microbiology lab in high school, one of the techs told me just flat out, if you grow anything from a urine sample, it means you're sick, because that urine has been filtered from your bloodstream by your kidneys. So anything that comes into the bladder is going to be some kind of serious condition that you need to be looked at. So, and with that kind of foreshadowing, the bladder is not sterile. Outside of an acute infection, there's a stable population of bacteria that just loves living in your bladder. Some people don't have a microbiota in their bladder, but the majority, in this study, the majority of women they studied did. And this is the work of Alan Wolf and Linda Brubaker. They found the evidence of uncultivated bacteria, and then later they actually did cultivate bacteria in a different study. This leads naturally to the question about what this microbiota might be doing to the human host. From the Human Microbiome Project, they discovered, they've been working on other body sites and discovered some uses that the microbiome can do for the health and for disease to the human host. Some guesses extrapolated from those studies would be, as far as like good things it could do for a human host, is to be a physical barrier to pathogens. They could outcompete pathogens. They could promote proper cellular development of the entire urinary tract. That's a big surprise. They could degrade harmful compounds that are introduced into the bladder. These are the good things that might be happening. Of more pressing interest are when bad things happen. And for this example, the urinary incontinence is a affliction that is complicated and is still an active area of research. Recent key findings have been that an altered urinary microbiome with urinary incontinence was discovered in compared to asymptomatic women. Greater symptom severity was discovered with lower diversity in the microbiome of the urinary bladder. And then a relationship was observed between the response to treatment and the diversity of the urinary microbiome. So with that, a common method to get to these conclusions would be to sample bacterial populations from as many different conditions as possible, and then make correlations between the populations that you've discovered or populations you've identified and the state of the health of the host there. Key to this is accurately identifying the bacteria in a sample that you've obtained. And for this, the sequence of the 16S RNA gene, ribosomal RNA gene, is commonly used, but it's not always the best method. So with that, identifying bacteria is difficult. They're hard to see and they're hard to characterize. There's been a long history of frustration to exactly agree on what kind of criteria to use to identify, to demarcate one species from another. With the advent of sequencing technology, this became easier. It's still not foolproof, but with the entire genetic information from a bacteria, you can go a long way. And this is practically the gold standard, is that you take all the genetic information from bacteria, sequence it, and then compare it to known bacteria and see where it would lie on that tree of life layout. The benefits of the whole genome sequencing is that obviously you get all the information you could possibly get, and then it also allows you to predict the functional profile of what the capabilities of that bacteria based on the sequence. There are two ways that you can get the DNA to actually do the sequencing, and the first one of the ones is the most reliable one, is to actually grow it yourself. Clonal populations give you a large number of exactly the same genetic material, and sequencing that doing a whole genome sequencing on that gives you a higher confidence for the classification of the bacteria, and there's no amplification bias. Cons for this is that the assembly and annotation of these genomes are difficult and computationally intensive. Optimizing the culture conditions can be very difficult, and this was realized early. By 1932, they were already publishing about how hard it was to grow bacteria of a specific interest. A lot of work has been done since then, but if you can get around it, it's desirable. And lastly, sequencing an entire genome is expensive. The next strategy you get for getting the DNA is to just extract all the DNA from the sample and sequence everything. For human microbiome samples, this naturally includes all of the host DNA. Or if you're sampling dirt, you get all the DNA from other organisms that are not of the bacteria of interest. Good news is that you do capture a representative of the environment, both the known and the unknown bacteria. The cons, obviously, well, not obviously, but the biggest con is that there's a lot of wasted effort due to the human contamination from a human sample, like urine, and it requires a high depth of coverage due to the fact that most of the biomass is going to be your host. Filtering that out leaves you with sampling biases or losing all the DNA samples that you could get from the bacteria that you want. And again, finally, sequencing an entire genome is still expensive. And the last technique I'm going to explain here is the polymerase chain reaction. It's been around for a long time. It's robust and fairly straightforward. The benefit of this is that there's no assembly or reference mapping required if you do the right technique. If you're just looking at the specific amplicon that you're sequencing, you're not required to use a reference genome to map it against. Again, choosing the right target means you can circumvent the entire host contamination problem. There's a low coverage requirement. It's relatively inexpensive. You still have to pay for the sequencing time, but you can fit a lot more samples onto your run than trying to get one whole genome. The cons is that it's reduced resolution for identification. Necessarily a smaller piece of DNA holds fewer information, or less information than would the entire genome. It does not provide any kind of functional genetic profile, and the polymerase itself introduces errors that need to be accounted for. So with that, do you need the entire genome to make an identification of a bacteria? The answer is no. You can use smaller parts of informative DNA that are not the entire genome, and genes are a good start. Can you use any gene? And the answer again is no. Bacteria have an alarming ability to share genetic material that works. They're sharing tools between themselves. They can do this in three ways. They can do transformation, which is literally absorbing DNA loose in the environment into their body, into their cellular structure, and that works. They do have a particular extra chromosomal genetic entity called a plasmid, which directs dedicated structures to pass genetic material between themselves. This plasmid is actually getting passed from between the donor and the recipient. And then finally, bacteria get sick too. Phages will inject DNA into the recipient bacteria, and sometimes this will actually confer benefits to the bacteria. This is all considered lateral gene transfer. In the literature, you might come across the horizontal gene transfer, and like I just demonstrated, I always trip that up. So I'm going to say lateral gene transfers from now on. And the downside of this is that choosing a gene that gets passed around through lateral gene transfer means that identifying two bacteria could be confused because they're sharing the same gene by passing between themselves. It shows no real taxonomic resolution. The sequences can be still very similar, and that's not a good way to choose a marker gene to identify bacteria. And, however, some genes are less likely to function in the recipient bacteria, and those are the ones you want to use as marker genes. Early on, it was identified that genes related to transcription and translation are less likely to work from lateral gene transfer. They're resistant. Not resistant as in like, I'm resistant to disease, but the physical act of transferring this DNA into a recipient bacteria and having it function in that foreign body is less likely to happen. So in this picture, this is a DNA strand right here, and on it are RNA polymerases that are transcribing messenger RNA, and on this are these huge beads here are the ribosomes. They just stack up as soon as there's enough room for them to start translating into protein. In the 70s, Carl Woese identified that ribosomes are shared in all forms of life, and this would be a good way to determine relations of the tree of life using that specific characteristics. First, he did digested fragments of ribosomes, and later, once sequencing technology got good enough, they went straight to the gene sequence. So this is it here. This is a picture of the ribosome. It's got two subunits. This is a large subunit and a small subunit. It's got several different. It's made up of several different materials here. You've got protein, which assists the folding of the ribosomal RNA, and then inside there you've got some transfer RNA, and then at the end here is a little residue of the amino acid, and I'll come back to that part later. For this ribosomal RNA here was used by Carl Woese and commonly still today as a reliable method, for the most part, to identify bacteria. This is the secondary structure of the ribosomal RNA of E. coli. And I'm going to spill the beans here. This red part here, this is the V4 region. This is a smaller part of the entire sequence, and this can be used reliably to identify bacteria, and this is commonly used nowadays for the bladder microbiome. So in summary, a good candidate marker gene is one that's likely to resist lateral gene transfer, and therefore the differences between the gene sequences are due to mutational rate. There are genes present in all bacteria, and at least the environment of interest. There are studies where they'll find homologs in the environment of bacteria they expect to find, or they'll sample the bacteria of that area and compare the genomes and find the genes that are good for that specific environment. And then the next one is single copy. Some genes are present in multiple copies in bacteria. The 16S gene is one example. And then finally, a good quality is that these proteins are part of larger complexes. So now I'm not discovering marker genes on my own. There's been work, and the success of these two studies has left us with 40 protein encoding marker genes that can be used to identify bacteria. For this paper here from Francesca Ciccarelli, she used the computational method of comparing thousands of known genomes of bacteria and picking out the genes that were present in all of them. And then later, Rodham Sorek did a laboratory validation of these marker genes. And from his laboratory work, he showed that these genes are resistant to being laterally transferred between bacteria. So I'll be using this list in my work. So what are some examples of these? For the most part, the 40 marker genes are involved with the ribosome. And the majority are these proteins that guide the correct folding of the ribosomal RNA. There's over 20 here. These are all good candidates to use to identify bacteria with. The next class are the specific enzymes that attach the amino acid residue to the transfer RNA. They're called tRNA synthetases. There's two classes. One class is a small enzyme, all in one piece, that will do the attach the amino acid residue. Those are not good for marker genes, and I've already weeded those out from the list, but these are known. The next class is what the important one is. Oops. This is the synthetase here. It's got two subunits. This pink part here is the transfer RNA, and because these are multi-subunit complexes, this is a better candidate of this class of molecule. The next two that are used are the alpha and beta subunit of the RNA polymerase. The blue parts here are the alpha and beta subunit. They're clamps that hold onto the DNA strand as it's transcribing messenger RNA. And then finally, there's like a small collection of associated enzymes that will charge the ribosome with energy, or in this case, will move unfolded protein across the membrane into the periplasmic space here. So for this specific marker gene, it'd be the sec-Y right here, this green part. Okay, do you need the entire gene, though? Not always. You can pick smaller parts of the gene. There are more informative parts of just that gene that can be used. And for an example of that, I've got, again, the secondary structure of the 16S RNA gene between bacteria, the bacterial domain, and the archaea domain. And these orange parts are the variable regions, or the parts that are variable. The V4 region does cover a larger part of this, but these are the important ones here. So if you would take this, these RNA sequences, actually the gene sequences, this is the transcribed RNA, but the gene sequences are reflective of this difference here. I can compare this part with this part, and I could tell from at least, I'd say all these, that this is bacteria, and this is an entirely different domain of life. So with that, I can go to my specific aims now. So my first aim is to find single variable regions from marker genes and compare them to how successful they identify bacteria from the human bladder to the V4 region of the 16S RNA gene. This will be a one-to-one comparison from all 40 genes, from the culled down list of 40 genes. I'm removing the synthetase molecules that are prone to lateral gene transfer. But of those 30-some marker genes, they will all have variable regions in them. And I can take each one of those and compare it to the V4 region of the 16S gene by itself. The second aim is to compare sets or combinations of variable regions to the V4 region by itself. And then lastly, it'd be nice to see if this actually works in the real world, so I'm going to go ahead and do it. After finding out which marker regions are going to be best compared to the V4 region, I'll design primers for them and then amplify DNA extracted from urine samples from Lisa Carson's lab. The sequences are important. I can't just do this out of thin air, and I don't want to use genomes that are related to other parts of the world. I don't want to use bacteria found in dirt, even though they are universal marker genes. Lucky for me, Crystal Thomas White and Alan Wolf have actually cultured bacteria out of human urine from human bladders and isolated them and then sequenced them, the entire genome. This is fantastic. I'll be using these gene sequences to find my variable regions based on these bacteria that are specific to the human bladder. So what they found was 149 isolates. These cover 78 different species, and they are from a representative of just 3 phyla of many in the bacterial domain. So how to do this? I'll be constructing multi-species alignments. And of those, I'll be looking for parts that are not conserved. Those will be the variable regions. And then doing my first and second specific aim, comparing these regions to the V4 region of the 16S gene. And then the last step is to validate these identified variable regions on urine samples, which is to design the primers, extract DNA from urine samples, sequence the amplicons, and then assign taxonomy. The method is based on multi-species or multi-sequence alignment. This is a difficult problem. Aligning 2 genes to itself or 2 sequences to themselves is solved. It's a little bit of work, but it's not as difficult as trying to align many different sequences together. And nowadays, work typically involves aligning like tens of thousands of sequences with each other. There are specific algorithms that do this kind of work, and they're a big advancement. I'm not going to go that detailed. There is a alignment algorithm that was made for small groups of multi-sequences, like 100 sequences or less. I don't have that many. I have about 78 sequences to compare. So this is a good fit. The method, trying to align all these sequences to themselves all at once is extremely difficult. So to get around this by doing a heuristic of doing pairwise alignments, all possible pairwise alignments, and then building a guide tree. And then those sequences that are the most identical to each other are aligned first, and then the increasingly less identical sequences are added to it until it's progressively added until the full alignment is complete. For this algorithm, it's called T-Coffee, and it was designed by Notre Dame and colleagues. In their paper, they used the example of these 4 sequences here. I'm sure you've read it by now. So pairwise alignment, or progressive alignment, without in the first attempts suffered from aligning sequences that had fantastic pairwise scores, but were not necessarily the best for the full multi-species alignment. So errors that were introduced early in the alignment were compounded until finally the final sequence, multi-sequence alignment, was not the best case. Those steps are to first do global alignments of each possible pairwise alignment. And in this example, there's the four sequences lined in the 6 different ways to each other. And then they're scored for percent identity. And in this scoring method, mismatches count against you. But these dashes here, these are called insertions or deletions, or just grouped together as called indels. These don't count. So even though this is obviously not, 100% identical to each other, there are no mismatches. So that weight, that score is 100%, and these are less. So after that, a guide tree is created, and they just work their way down. They start with these 2 sequences, then add sequence C, and then add sequence D. At the end of it, the problem is that you can see here, just visually, you can just push this over to here. There's a strong urge for me to just push this over to this side, and it'd be a better alignment. This is the downside of progressive alignment without any kind of consideration about the big picture. T-Coffee adds a consistency method to this. So again, it starts off by making all pairwise alignments, and then it goes through every possible combination and looks at how consistently each of these letters are aligned with the other corresponding letter. And in this case, the word "the" is consistently aligned with each other. For something like fat and fast, it's maybe not quite as consistent, or sometimes not at all. Sequence D has no information to give about Garfield, so that isn't included in the weight. At the end of it, you have a list of weighted values that will inform a dynamic programming. So as this dynamic programming is going through the final alignment, these 3 letters will be consistently will be weighted to align against each other. And over here, these weights are smaller than having fat aligned with fast. And at the end here, you see that it does a really good job at making a better alignment. So I'll be using that. The good news is that the good part for me about that is that T-Coffee outputs 2 files that I can use. One is the actual alignment, and the second is the guide tree. So, for example, here is a multi-species alignment. Each sequence from the Thomas White data set is aligned along the rows here. This is the 16S RNA gene sequence. It's the full length. It goes from 0 to a little over 1,500 base pairs. If you look at it by eye, you can see that, so the score of the position here, what are lined up at this position, the residues lined up at this position here are almost identical. And here's an identical stack of residues, and here's an identical stack. And when you get these blank spaces here, these deep valleys, that means that they're not matching, or there's big deletions or insertions there. If you just look at it by eye, which I did at first, you can kind of see that here's a group here, here's a group here, here's a group here. These are pretty obvious variable regions. These are not. I wouldn't have picked those out on my own. So doing it by eye is not a good idea. These variable regions have been mapped out on the E. coli genome, and I'm using those map positions here on this diagram. So this is variable region 1. This difference here is due to the fact that the bacteria that I'm aligning are specific to the bladder. Whereas otherwise, if I'd used like all the genome sequences on NCBI, it'd be a different alignment score. There are 9 regions mapped out, and they match pretty well. And just as a note, this is a T-coffee multi-sequence alignment. I did do it with Cluster Omega, which is the progressive aligner that can do tens of thousands of sequences. The downside with that is that it does lose some accuracy, and I think it was the V6 region here. There was a giant dip right there, and with the T-coffee, that's not included there. So that's good news. So to identify these variable regions without using my eyes I'm going to let the computer do that work. So the way to do that, the method to do that is a sliding window analysis. So this is the output file from T-coffee of the multi-sequence alignment, and this is not. I'm going to take a window of 200 base pairs. This is not 200 base pairs here, but it wouldn't fit on the screen. So this is the window. 250 base pairs is a ideal length for the MiSeq sequencer platform, and I'm going to use the score of these positions to sum up a total score for this window. And in this position here, I've got a high score. And as this window moves along stepwise, and I add up those scores, they're going to get worse and worse and worse and worse until I bottom out here. And then as I move along further to a part that's conserved, that score is going to get better and better and better and better until finally I'll get this. This is the ideal situation here. This is the variable region that I'll identify. And the best case is that it's flanked by two completely conserved, 100% identical regions. That's the best case for designing primers. I don't expect that to happen all the time. So the next step is to do the comparison of the variable regions. To do that, I'm going to use the guide tree that's generated by T-coffee. So this is the full guide tree here. This is the map that T-coffee uses to do its to build the multi-sequence alignment. And on there, this is being generated by a pairwise similarity. It's got like the most unfortunate acronym ever, I think. It's unweighted pair group method with arithmetic mean. I can't even pronounce the acronym. It's horrible. But what it means is that it uses a distance metric to cluster sequences together until finally you get this guide tree. And the branch lengths here indicate how far apart, how different those sequences are. So here, a branch length of 0 means those two sequences are identical. And here are three identical sequences here. They happen to be strains of the same species. So that's to be expected. This is preliminary data, and I included strains inside this first multi-species alignment. For the final project, I'll have to curate that to make sure there are no strains, because I'm not looking to get strain level resolution out of this. And then here's another branch of strains of the same species. These are to be expected and ignored. So this is what I'm looking for. These two species have identical V3 sequences. I forgot to mention, sorry. This is a multi-species alignment of just the V3 region. And for this, the V3 region cannot tell these two species apart from each other. This is bad, but I expect that I don't expect one region to be able to identify all the species of bacteria in a sample, which is why I'm also going to be doing comparing multiple variable regions with each other. So of that, I can just count these up. And then from that, I can tell how many species each of these variable regions correctly identified. So here's the gold standard, or the standard so far is the V4 variable region. It identified 69 of 84 species correctly. That's 82%. And then compared to these three other variable regions, V3, V6, and V8, they all managed to identify more species correctly, and they have a higher percentage here. You'll notice that these are tied, V8 and V3. They identified more species than the V4 correctly, but not the same species. And that's in this slide here. So this is the final, this will be the final step in this method, which is to find the minimal set of variable regions that can cover the entire species population in that sample. And for the 16S gene, for each of these four variable regions, Staphylococcus is difficult. It consistently confuses these two species with each other, and for the V4, it also adds this extra one down here. Veillonella is another difficult one for them. For all four of them, Peptonephilus is really difficult to determine, and then these others are a smattering of other genus level identification. So genus level is not bad. I'd like to get species. That's the goal, but genus is okay for now. And then again, up here are the two unrelated species that are confused with each other, which I'm going to have to weed out. So it might be that from this, I would say, I don't know, if I had to pick two of these, I might pick like the V8 region and the V3 region, and then look for other marker genes to fill out the rest of the populations. And then finally, to do the validation, we'll be designing and testing the primers on extracted DNA, sequencing amplicons, and then assigning taxonomy. So as I said, the best case, these variable regions will be flanked by completely conserved regions. In the case that they're flanked by regions that are not completely conserved, I'll be using degenerate primers, and I'll use code hop for that. So for this diagram here, that means that for all those segments that are different from each other, I have to design one primer to be able to amplify that section. So of this, I'll need roughly, for this example, it looks like there's 16 different primers that are necessary. So it'd be ACA, ACG, blah, 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 and so on, until you get all 16 different primers to use in that set. This is the degenerate part. But code hop is an established way of doing this. And then finally, to assign taxonomy, I'll be using ID taxa, which I can train on the Thomas White data set, and it assigns a statistical confidence level to the taxon assigned to it. This is how long I think it will take, August. With luck, I'll be done. And then that covers everything. I'd like to thank my mentor, Lisa, and my thesis advisory committee, which is Mark and Michael and Holly, and then everyone else at DMICE, Christina Zhang for questions I had about sequencing, the Carson's lab, Ian, all listened to my presentation more times than they care to admit, and then the Asquith lab for, I'll be eventually using their materials and their space, and I'm going to thank them in advance. So, thank you.
Thank you for sharing that. I was really glad you showed us some preliminary data, because the question I thought about at the beginning when you started was, would you be able to do specific AIM3 if nothing performed so well in specific aims, 1 and 2, as to be identified as being better candidates? So I'm a little reassured because you've shown some evidence that you think they will be better candidates, but I'd love for you to talk a little bit more about how much better do they have to be to kind of meet your definition of being better candidates, and to make it worth doing the specific AIM3 differently?
Yeah, okay. So, I'm trying to, yeah, I think once I have this in front of me, I'll be able to see a clear pattern. There is a little bit of nervousness because these sequences are only 300 base pairs. For this work, what they did was they used they did the full genome sequence, and then they used the 40 marker genes. They just pulled out the 40 marker genes sequences and concatenated them all together. So they've got 40 times however long those genes are, and then they use that to match against a database. That's not an entire genome, but it is still a lot of information, and I'll be using even less of that. Hopefully more, just as informative though. So I think it would be if they're reliably distinguishing between a set of species, and then even if they are not reliably identifying the other 140 isolates, I can still pick up some other variable region from a different marker gene. Hopefully it's going to be a small set. There could be, I haven't looked about how many variable regions are going to be in these functional marker genes, the ones that encode proteins. It could be, I don't think it'll be 9 per one, but maybe 5, so whatever, 5 times 40, still a lot. So I'm hoping in those numbers that I'll be able to find something that will cover the entire set reliably. But yeah, as far as an exact threshold, I haven't got to that point yet.
Hi.
Hi.
Thanks for an interesting talk. I do have a couple of questions about the details of the methodology which weren't clear to me. It sounds like, and I'm probably wrong, but when you're looking at whether you can distinguish one bacteria from another, you're looking at like any difference in the base pair window at all. Is that the case, or is there some threshold of difference or some sliding scale of difference that you're using? And the reason I'm asking is because, you know, this is not my specialty, but it would seem that when you translate this into its eventual, you know, clinical goal, which is some, you know, fast clinical tests for doing this, that more distinction would be easier to measure and easier to build a microarray chip or whatever it is. So just, you know, what are your thoughts about, you know, how much difference that you really need, or how you will measure how much difference is really necessary?
Right. So for example, of this, for this I picked a threshold of 0.025, and that corresponds to roughly a 5% difference between the sequences. This is not assigning taxonomy though. This is a threshold I picked just to use as a degree of similarity to those sequences to each other, those short 300 base pair sequences to each other. I'm depending on ID taxa for the final confidence level about what threshold I should be using. So for this, when I was just eyeballing it, there seemed to be a clump where you had 0, and then a few more values above that, and then a large gap of empty space, and then everything else was larger values corresponding to larger distances. So of that, I'm going to be looking at exactly how this data clumps or clusters. Be looking for having the data tell me the threshold I can use to reliably separate these things out.
Yeah, I wonder if there's like a multi-objective optimization in this, as you want to both separate everything as well as separate it by the you want to cover everything, so right, but you also want it separated by as much as possible.
Yeah, and that's what the variable regions will be for. Those are the parts of the gene that are most different from each other. I don't expect it to be as stringent as a single base pair difference. That's not. I can't expect that. But then again, I can't just say, anything over 10 base pairs is going to be something is just out of consideration.
Do you have an idea, just looking at again the practical end aspects, what level of difference is, that people would be confident they could create probes and stuff that would be reliable, and use that sort of as a minimum threshold? What would be practical in the long run, kind of thing?
Sorry,
just like we're trying to separate this. So when with the eventual technology that's going to use this, how much difference do you need for that technology in the end to be reliable? Whether it's DNA probes on a DNA microarray or whatever it is. I guess it'd be RNA microarray in this case, but one base pair doesn't seem realistic, right? 'Cause,
Yeah, realistic.
You know, partial binding and all that kind of stuff. But, you know, 1,000 seems way more than you would need. So, you know, what is the, you know?
So for this, it's just, I'd be looking for the amount of differences in those variable regions. I don't know if you can, I wasn't considering like a step after that, is it like making probes to these specific regions? Any kind of DNA probe that I'd be making would be the primers themselves, which are actually going to be located outside those spaces. But yeah, I would have to, I'm expecting to look at what I pull out of this data to see if there's a concrete way of determining a threshold for that.
So one other little question, not related to the first question, is how do you know what the answers are to the gold standard? You have these urine samples. How do you know what's the right answer?
I don't. I have some expectations. I expect that this is slightly akin to like a training set and a test set. The training set would be these sequences because they were extracted from bacteria in the bladder. So my expectation is that having amplicons specific from this or built from this data set is going to be able to also identify bacteria in an unknown data set of urine samples. I would not expect to find things like deep sea vent bacteria sequences in there. But that's what the statistical way of doing the taxon assignment would be for. Using ID taxa to train this data set on the identification of those sequences would give me a confidence about what I'm looking at in the unknown bacteria, just how confident can I be saying, yes, these bacteria are in it. As far as the unknown bacteria that I don't know are in that population, I really have no way of saying if they're there or not.
Right, but isn't there always at least the possibility that in the unknown bacteria, their sequences look like some of the known bacteria, so you get, you know, kind of a false positive. I wonder if there's any way to, you know, improve that by, you know, also looking for similar things that you don't expect to be there and somehow showing that they're not there. I mean, I don't know. It's always difficult when you don't actually know what the right answer is in a new sample. Or, I guess the standard answer would be to use some other more laborious laboratory technique on some subset of the
That's what this
samples
is. They actually grew it out there. And the answer to that would be to take more samples from more bladders and extract the DNA and do more extra culturing on that.
We also could see if they still have the urinary incontinence one, and be able to do
Oh, yeah.
the marketing
Yeah.
if they still have the development.
Right.
Which is possible.
Cool. That's it.